For the scientists that listen to your podcast, I would ask them to not only think about their slice of the pie, their slice of the value chain, but look to the left and to the right. Who's going to take the output of their work to the next phase? What do they need? What does good look like for them and for the next step and for the next step? Because not all of us want the same thing, which is to have these products impact patients positively. These silos, the traditions of scientific research are holding us back today rather than enabling us to really achieve that big outcome.
Welcome back to the Smart Biotech Scientist. We're continuing our fascinating discussion with Jason Foster, who is the CEO of Ori Biotech. In part 2, we're exploring tech transfers, scale-out strategies, and explore why scientific excellence alone does not guarantee success. Let's dive right in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
Now, I want to look at another aspect because we have different molecules, we have different cells, different cell types, and we have also, very importantly, different volumes in development, or even when you're producing for different patients, the volumes can vary a lot. So how do you tackle that with the Euro?
So the way the system has been designed is to be cell type agnostic. So we've worked with CAR T cells, TILs, TCRs, NKs, dendritic cells, all kinds of different cell types. So anything that expands or exists in a suspension context, suspension culture, can be done in the system. And also we built it for flexibility, because part of the challenge that exists today, there's been systems that have existed for over a decade, and they are often used in a lab scale context and very difficult to then go into a GMP scaled up context, but they're known for being inflexible. So you have to be able to shoehorn your process within the constraints of the technology. What we were trying to do is to build a technology that could flex to the needs of your process. And so that thinking is fundamentally different. In the single-use bioreactor consumable that goes inside the Eero instrument, you can operate at 50 milliliters. So you could do a unit operation like you might do in a T-flask, a genetic reprogramming step or an activation step potentially, and work all the way up to 1 liter. So you can go 50 mil to 1 liter and anywhere in between. So we can start with 50 million cells, patient starting material, and go to over 12 billion cells of therapeutic product in the same vessel. So that's how we've overcome some of that. We'll have to go from a flask into a bag, into this piece of automation, and maybe back into a bag, and then into this other. That's what today's process looks like. But if we can go from small volume to high, and we also can do static mixing effectively, that the cell culture can sit as it might in a T-flask in an incubator, or we can also rock the culture, and we can also then mix in a different way, which is called compression mixing. So we have the ability to do static or active, low volume or high. You can change speed, you can change amplitude, you have all those parameters in your control. And this is part of what one of our customers said to us is like, I view the Eero as empowering scientists to use their expertise to be able to solve these problems and develop the best therapeutics, because the tools they have today are relatively inflexible. They don't have many levers that you can pull to try and solve problems. It just does what it does. But with the Eero system, you can actually think about, oh, actually, I might want to do it this way, or I might want to do that faster or slower, or at a higher volume or at a lower volume, or I might want to interject this other step in between. And that empowers the scientist to really solve these problems in new ways, which I thought was a great way to think about it.
So can you do both suspension and adherent cell cultures?
So the bioreactor consumable we have today only does suspension. There is a version of it that will do adherent. We just haven't brought that to market yet. It's a much smaller part of the market today. So, I mean, you hear a lot about iPSCs and HSCs and MSCs and macrophage, but it's a very, very small part of the market today. And so we're just waiting for that kind of part of the market to hit critical mass. But I think it's something like 80% of the cell therapy programs are T-cell based, CAR T or other. So that's where we started our work partners.
And where do you see the current limitations with respect to comparability or also say control, like the digital side or scale up? What are the challenges you want to still solve with that system?
We're excited about the opportunity to get into the clinic, hopefully in the second half of 2025 with a partner. So to get the first patients treated with and demonstrate the use of the product clinically or the platform clinically. And then we can think about, well, how do we go from clinical scale to commercial scale? How do we go from 10 doses a year to 10,000 doses a year? And that's really the ambition and what the platform was made for, is that we wanted to be able to do tens of thousands of doses a year. We've got large indications, cancer indications that need that many doses. We've got autoimmune in focus for CAR T, leaders like Kyverna and Cabaletta and others that are doing research in lupus and ALS. Lupus itself is 50,000 patients a year. Altogether as an industry, across all the products that exist, we might treat 10,000 in 2024. So we need a step change in the quality, the quantity, the throughput of these systems. And that's really what the Eero has been designed for, to use the same platform to do R&D as you're going to do for GMP manufacturing. So same instrument, same consumables, same process, which will smooth the transition into the clinic and to commercial scale. We think actually because shortening the tech transfer time, lowering the comparability risk by using the same platform, we will shave about 3 years off the drug development timeline for a cell therapy.
Yeah, that's massive. That's a lot of savings.
Yeah, it has huge cost implications, and obviously it's great for patients, getting products to patients more quickly. So we're excited to see that part of the journey start to unfold as we get into clinical trials next year.
That's amazing. And how will the scale up look like, or should I say scale out? So is it the same size and you will have several ones, or you are envisioning also much bigger bioreactors?
No, I think the way our approach differs from the biologics approach is we don't scale up volume, we multiplex. So you have multiple Eeros operating at the same time. So in parallel, we can manufacture for 30 patients in one clean room. So about 1,000 square feet of clean room space. We can deliver 1,000 doses a year. And that would include 30 patients in parallel. So the ability to actually, because it's a personalized medicine, we can't obviously mix the cells for 2 patients, or people are talking about allogeneic, and allogeneic could be something at some point. We know that there's complexity around the safety and efficacy, durability, and potency of donor cells. Thus far, we haven't been able to overcome that hurdle really. And that may work at some point. But I think right now, we just can't have donors like plugged into the matrix constantly donating T cells. It's sort of like there's going to be donor to donor comparability issues. There's going to be all kinds of things that might cause some challenges. So what we're thinking about is how do we just run in parallel an individual patient's dose next to another patient's dose, and we optimize throughput that way. So this idea of mass personalization, being able to maintain that personalized nature, but have it be done at a large scale. And that would be achieved with the Eero system through a multiplexing. So essentially stacking the instruments up and being able to intensify the footprint. We think based on the square footage required, we could reduce the amount of clean room space you need by about 95%. So
wow,
it's a massive reduction to deliver the same number of doses. The overall facility size, because there's so much other things happening besides just the GMP manufacturing piece. There's warehousing, there's office space, there's all these other things. The overall facility would shrink by about 50%, but it's still going to cost $150 million to build a massive facility. It's important that you're only building what you need and you're getting the optimal throughput and the optimal utilization of that throughput, which is today a big challenge.
You're making a good point here because, I mean, if you don't need to build a complex facility or you need less clean rooms, you can save a lot of capex. But then also in the long run, you can save a lot of operational costs. I mean, cleaning costs, maintenance costs, and so on. It's a huge saving.
It is. I think the numbers that I heard quoted was it's about $1,000 a square foot to build. So if you need 150,000 square feet, it costs $150 million to build. And I've seen these facilities twice that big, so $300 million to build. And then it's about $30 million a year just to keep the lights on, just operating costs. And that's significant. And especially oftentimes if you're building a bricks and mortar facility, you have to put a certain amount of capacity into the facility. I'm going to build a certain footprint that can do a certain number of doses, but you don't start out at full utilization. You start out very low utilization and hopefully you work your way up. but you've got a lot of underutilization and you're spending tens of millions of dollars to keep the lights on while you're not running revenue through that facility delivering doses. So we have partnered with a couple of the leaders in the mobile cleanroom space. So GCON, Germ-Free, Ardmac in Europe to create what we're calling the factory pod solution. Effectively, it puts the Eero technology inside of a modular manufacturing space, cleanroom space, and that is pre-designed. So essentially, it's a turnkey manufacturing setup for cell therapy that has the ability to go from preclinical development to commercial scale. And that would preclude the need for a giant facility. You could have a couple of these and potentially you get to a certain amount of capacity for one, and then you put another one next door and then another one next door. And you have a de-risked scale up. So you don't have a bunch of excess capacity sitting unused, which is a very expensive thing to have at the beginning.
Yeah, definitely. Now I'd love to tap into your commercial experience a bit more. And I want to ask you this question. What do you see as the key ingredients for successfully translating innovative scientific technologies into commercially viable products? And why does scientific excellence alone not guarantee success?
Well, we covered off a little bit on this at the very beginning. I think that is the billion-dollar question of why some of these products haven't achieved great commercial success. And I think we're just starting to see, yes, Carta, I think, is the first blockbuster cell therapy product, but they have this high cost of goods that everyone else does, and they have the challenges with throughput and out of specs that everyone else does. So I think it really fundamentally goes back to elevating commercial viability up to the same level as safety and efficacy in the preclinical development process. We need to know, is the therapeutic that we're developing, does it have the right impact? Does it have the right safety and efficacy profile? Are we getting that signal that we're going to move into the clinic? That is all that has mattered for the last 5 decades. Ever since we've been manufacturing pharmaceuticals, we've only really cared about safety and efficacy because we knew, certainly with small molecules, that we could make it. If we could get the chemicals to combine in the right way, we could stamp out tablets. We've been doing that for years. Biologics, we started out with some of the similar challenges that we have today with cell therapy, but we figured that we overcame them. It was always assumed that manufacturability was just a given, that we could always manufacture so long as we could get safety and efficacy. Well, our history in cell therapy over the last 7 years has proven that that's not the case. And so that you can have very safe and very effective product that either never reach patients because the profile doesn't match up. Recently in the UK, a company that we know very well, Achilles Therapeutics, had 2 products in the clinic that were getting good results, good clinical results. And ultimately, the board and the management team decided to shut the company down because they realized that those products were not going to be commercially viable. The process that they had created to make those products was never going to be a point where they could deliver an ROI and where they could make enough of them. And I thought that was a very interesting outcome for that company. That I'm sure it was a very, very hard decision for the management team and the board. But to say, they still had something like, I want to say $90 million in the bank, publicly listed company. And they said, you know what, these products aren't commercially viable. Now, this is after they had been in the clinic for several years. So if we can move that decision point earlier to say, what are those viability points that we have to be able to hit? So I'm an old school pharma marketer. I remember what a target product profile was back in the day. Like we had a TPP that says, okay, it's got to deliver this clinical benefit. It has to have this safety profile. It can't have these adverse events maybe, or not to that extent. But we never used to put in there a cost of goods because when you're stamping out tablets, the cost of goods is a fraction. It's the $1 billion you spent to get there, but the cost of goods isn't even relevant. For cell therapies, it is relevant. If the cost of goods is too high, that means your price has to be even higher to make any money. That economic profile of that product isn't viable. So you have to say, okay, we need to spend the time working to lower the cost of goods so we can actually get the right balance here and get a product that can be covered. But that needs to happen before you hit the clinic, because by the time you hit the clinic and certainly phase 2, 3 or the late stage pivotals, you're not going to change anything about your process. The process itself will be fixed. Therefore, the costs and other things associated with that will be fixed. And you could very much, unfortunately, end up launching a product, getting approval, but the product will never be able to be covered and never be able to treat patients. And that is the worst case scenario for everybody. You've spent the $1 billion to get it approved, but the patients can't get the benefit. The economics don't work. So we need to pull all of that forward. And my suggestion is that we elevate viability up alongside safety and efficacy, preclinical, to make sure that we've got a product that can reach patients and can have the clinical and economic impact that we require.
Yes, this is so true. What point of having a drug that could save a lot of patients, but we cannot manufacture it because it's just too expensive? It's just not possible. And especially, it's sad that they came to that conclusion very late because it's years and years of energy and time spent and money spent.
I would argue that there are many companies that actually take it all the way through, and they never come to the realization until after they've commercialized that actually that product is not viable. So, I mean, kudos to them for making the decision before that. But you're right. It should be happening before you hit the clinic and before you spent those years and hundreds of millions to get the product to a point where it's ready to go clinical.
We're definitely in a space where we still have a lot of problems to solve, economic problems, scientific problems, distribution problems. And you say that you're not afraid to solve hard things, so that's good. You're in a good spot. Now, my question, Jason, is where would you love to see new innovations in order to be able to solve these problems?
I'd love to see the evolution of distributed manufacturing come to reality. I think the regulatory framework needs to be thought through differently, but it's absolutely possible. You need technology like the Eero platform, which is cloud native, it's automated, so you can replicate the same process in multiple places and you can control it and monitor it, which is really what's required to do this well. You need to have GMP standards no matter where you're manufacturing. So whether it's in 8 sites or 1 site, and that this platform and others will allow us to do that safely for patients and at high quality. And that also leads to other potential biological benefits. So if you're doing it at or near the hospital, potentially you can use fresh cells instead of frozen, and hopefully ultimately fresh therapeutic that doesn't have to be frozen, which has an impact on the cells, on the viability, on the potency of the medicine, and the durability of the medicine, and on the patient. So there should be some biological benefits as well, bringing manufacturing closer to the patient. The other thing that requires, or that kind of distribution model requires, is better analytics, better tools. Right now, we spend a week or two releasing a product, which just means like we have to test whether it's sterile and we have to do the release testing. All of that can and should be done in process, in line. So we should be able to know when the product comes out of the Eero system, whether it's met spec or not. We can tell you how many cells are in there. We can tell you ultimately, very quickly, we can tell you what viability is. We can tell you often what the phenotype or other things are. But a lot of that's done offline, and that takes time, and that takes money, and that's just time that the patient often doesn't have. So my hope is that we get better inline analytical capabilities so we don't have to do so much offline and after the end of the process. Because essentially the therapeutic is waiting. It's waiting there, ready to go back to the patient. But we have to jump through all these hoops and go through all these steps that aren't necessarily value added for the product or for the patient, but they're required.
This has been fantastic, Jason. As we are wrapping up, what is the most important takeaway you want our listeners to walk away with?
I'd love for them to really understand that this is an incredible new modality that could revolutionize almost every area of medicine. Cell therapies are being studied in cardiovascular disease, in type 1 diabetes, in all kinds of diseases that today are difficult to treat or impossible to treat. And so we're going to have to be flexible in the way that we approach them and the way that we think about these therapies. These are not small molecule drugs. These are not biologics. These are something different. But they're also not regen med, hospital-based therapies. We need to have GMP standards at scale for living personalized medicines, and we don't have them today. So for the scientists that listen to your podcast, I would ask them to not only think about their slice of the pie, their slice of the value chain, but look to the left and to the right. Who's going to take the output of their work to the next phase? What do they need? What does good look like for them and for the next step and for the next step? Because not all of us want the same thing, which is to have these products impact patients positively. These silos, the traditions of scientific research are holding us back today rather than enabling us to really achieve that big outcome.
Thank you so much, Jason, for sharing your insights, your passion, and for making a difference in this very important space. Where can people get a hold of you?
Very active on LinkedIn. Feel free to look me up, Jason C. Foster at Ori Biotech. And my phone number is on there. My email address is on there. If you want to reach out to me, that's a great way to do it.
Awesome. Thank you so much. I will leave the links in the show notes, so please do reach out to Jason. And Jason, thank you once again for being on the show today.
Thanks so much for having me. It's been great.
Thank you for joining us for this inspiring conversation with Jason Foster. His insights into the future of cell and gene therapy manufacturing remind us that innovation comes from tackling the hardest challenges head on. And speaking of challenges, are you a CTO facing seemingly unsurmountable bioprocess challenges? Let's transform those obstacles into opportunities. Book your free call at brühlmannconsulting.com. And thank you so much again for connecting. Thank you so much for your likes. Thank you so much for your reviews. And if you haven't left a review, please do so. It means the world to me. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.